In this lecture, we will examine the first part of oxidative phosphorylation, and that's electron transport. Oxidative phosphorylation is a process by which aerobic cells produce ATP, and it's composed of two parts: electron transport, which we'll examine in this lecture, and in the electron transport, electrons are donated by the reduced coenzymes NADH and FADH2 to the electron transport chain, or ETC. Electron transport occurs in the inner membrane of mitochondria in eukaryotes and in the plasma membrane of prokaryotes. Second part, which we'll examine in the next lecture, is chemiosmosis, and that's extraction of energy from electron flow and synthesis of ATP. The mitochondrial electron transport chain, or ETC, is a series of electron carriers arranged in the inner membrane in order of increasing electron affinity. They transfer the electrons derived from reduced coenzymes to oxygen. Coenzymes originate from glycolysis, the citric acid cycle, and fatty acid oxidation. Potential for NADH oxygen pair is 1.14 volts. That's actually rather high potential. If this reaction were to occur in a single step, a large amount of energy would be released, and that would be essentially uncontrolled reaction. That would be damaging to the cell. That's why energy is released in the electron transport chain, in which each reaction in a chain releases small amount of energy, and so energy is released in controlled fashion in a way that cell can use as a source of energy. The energy released during the electron transfer is used to facilitate endergonic processes. One of them is synthesis of ATP. Others include pumping of calcium into the mitochondrial matrix and generation of heat in brown adipose tissue. The components of electron transport chain are located in the inner mitochondrial membrane. Electron transport chain components are organized into 4 complexes, which are indicated as complexes 1 through 4, each of which consists of several proteins and prosthetic groups. In addition, 2 other molecules play a very important role. One of them is coenzyme Q, which is sometimes labeled as CoQ. Also, it's called ubiquinone or UQ. Coenzyme Q is actually a quinone ring with various substituents. So here is shown a basic molecule, CoQ, so-called CoQ0. In bacteria, side chain typically has 6 isoprene units, and it's indicated as CoQ6. And in mammals, CoQ has 10 isoprene units, and then it's indicated as CoQ10. Other important molecule is cytochrome C or Cy-C. Complex 1 is the NADH dehydrogenase complex. It catalyzes transfer of electrons to ubiquinone, UQ, or CoQ. Main sources of NADH are the citric acid cycle and the fatty acid oxidation. It is the largest protein component in the inner membrane, and it is composed of at least 25 different polypeptides. Complex 1 contains 1 molecule of flavin mononucleotide, shown here in 2 representations, 2 formulas, so flavin mononucleotide or FMN, and 7 iron-sulfur centers, iron-sulfur clusters. They are either in the form of Fe2S2, which means 2 or 4 sulfur atoms, so Fe2S2 or Fe4S4. So they contain 2 or 4 iron atoms that are complex to equal number of sulfide ions. Remaining coordinative sites of iron are bound to cysteine residues. Proteins that contain iron-sulfur centers are called non-heme proteins. In the complex 1, NADH reduces FMN to FMNH2. and then electrons are transferred from FMNH2 to iron sulfur center, one electron at a time. So if you look at reduction of FMN, we have two reactive sites. They are two nitrogen atoms that are part of carbon-nitrogen double bond. So they are basically part of a diene system. And reaction amounts to 1,4 addition to that diene system. It's effectively addition of hydride anion H- from NAD and proton H+. So H- and H+ add overall they add as hydrogen molecule, and they are shown in this equation as bold hydrogen molecules. Of course, that results in formation of new double bond in position 2, 3. If you recall organic chemistry, if we label original diene system, which in this case consists of two carbon-nitrogen double bonds, if you label that 1 through 4, then double bonds would be in positions 1, 2, and 3, 4. And newly formed double bond will be in position 2, 3, while two hydrogen atoms that have added in positions 1 and 4 are shown here as bold hydrogen atoms. Then electrons are transferred from FMNH2 to iron sulfur center, one electron at a time, and then they shuttle from one iron sulfur center to another, I believe there are 7 of them, until they are finally donated to ubiquinone, UQ. They are donated 1 electron at a time, so first after donation of 1 electron, UB-semiquinone intermediate is formed, and after donation of second electron, dihydroubiquinone is formed. Of course, donation of each electron forms corresponding alkoxide anion, which is protonated. And so donation of second electron, second alkoxide anion, which is protonated. And so the final product is dihydroubiquinone. Electron transfer is accompanied by movement of protons across the inner membrane into the intermembrane space, where proton gradient will be then used later to synthesize ATP that we will see in the next lecture. Complex 2 is succinate dehydrogenase complex. Complex 2 then consists of succinate dehydrogenase enzyme. This is one of the citric acid cycle enzymes, and 2 iron sulfur proteins. It contains covalently bound FAD. FAD, produced by the oxidation of succinate in the citric acid cycle, transfers its electrons to ubiquinone utilizing this complex, complex 2. So the role of complex 2 is to mediate transfer of electrons from succinate to ubiquinone to give dihydroubiquinone. And then that's transferred to complex 3 the same way dihydroubiquinone produced by complex 1 is, those electrons are transferred to complex 3. But while complex 2 transfers electrons to ubiquinone, unlike other electron transfer chain complexes, it does not transfer protons from the matrix to the inner membrane space. Complex 3 is the cytochrome BC1 complex. So role of complex 3 is to transfer electrons from dihydroubiquinone to cytochrome C. It contains 2 B-type cytochromes, one cytochrome C1 and one iron sulfur complex. Cytochromes are a series of electron transport proteins that contain a heme prosthetic group similar to those found in hemoglobin and myoglobin. Reactions are single electron redox or reduction oxidation reactions, and iron is reduced from iron 3+ to iron 2+. Ubiquinone is lipid-soluble molecule and diffuses within the inner membrane between the electron donors in the complexes 1 and 2 and electron acceptor in complex 3. Electron transfer begins by oxidation of dihydroubiquinone by iron sulfur protein in complex 3 to generate corresponding radical, UB-semiquinone, and next the reduced iron sulfur protein transfers electron to cytochrome C1, which then transfers it to cytochrome C. Complex 4 is cytochrome oxidase complex, and it catalyzes 4-electron reduction of molecule of oxygen to water. So molecule of oxygen plus 4 protons and 4 electrons gives 2 molecules of water. It is a membrane-spanning complex and may contain between 6 and 13 subunits, depending on the species. It also contains the heme iron atoms of cytochromes A1 and A3 associated with 2 copper atoms. Cytochrome A is associated with copper A atom, and cytochrome A3 is associated with copper B atom. Cytochrome C, which is located on the outer surface of the inner membrane, transfers electrons one at a time to cytochrome A and copper A. The electrons are then passed onto cytochrome A3 and copper B, which is located on the inner surface of the inner membrane, which already has oxygen molecule bound to it. A total of 4 electrons and 4 protons are donated to the molecule of oxygen, as shown above, to produce 2 molecules of water. And here is a summary of what we have just covered. NADH, generated in citric acid cycle or fatty acid oxidation, in complex 1 transfers electrons to generate dihydroubiquinone. FADH2, generated in citric acid cycle, utilizes complex 2 also to transfer electrons to form dihydroubiquinone. So, roles of either complex 1 or complex 2 is to transfer electrons to ubiquinone to generate dihydroubiquinone. In complex 3, dihydroubiquinone transfers electrons to cytochrome C, which then in complex 4 utilizes those electrons to reduce oxygen and form water. Each of those complexes has actually inhibitors associated with it, which we'll cover in the next slide, and they are shown in red. So in each step, electron gives up part of its energy rather than giving it all at once. So that way the process is controlled. And process actually occurs in 3 steps, from complex 1 to 3 to 4, or from complex 2 to 3 to 4. Commonly, 3 steps occur within complexes 1, 3, and 4, and somewhat confusingly, they are referred to sites 1, 2, and 3. So, these 3 steps that occur within complexes 1, 3, and 4 are referred to as sites 1, 2, and 3. And according to solid-state model, complexes 1, 3, and 4 form a supercomplex that is called respirosome. Respirasomes are found in animals, plants, and fungi, and is the most active and most stable form of the electron transport chain. Frequently encountered form of respirosome is a tetramer. It contains 1 complex 1, 2 of the complexes 3, and 1 complex 4. So it's 1, 3, 2, and 4 tetramer. Electron transfer is efficient because the active sites of different complexes are in close proximity to each other. And finally, we will look at few electron transport chain inhibitors. There are a number of specific inhibitors of the electron transport process. When electron transport is inhibited, oxygen consumption is either reduced or eliminated. And so here are some of the inhibitors. Rotenone is a secondary metabolite of some plants. It inhibits transfer of electrons from iron-sulfur centers in complex 1 to ubiquinone. Another inhibitor of complex 1 is amytal. It's a synthetic compound, a barbiturate, and it inhibits NADH reductase. Amytal was once believed to be a truth serum, because when a person is given amytal, he or she has tendency to talk. And so if that person is asked questions, he will answer the questions. So for that reason, because person will respond to questions and answer them voluntarily, it was believed to be a truth serum. But actually, while a person will answer questions and answers will make sense because there will be response to those questions, they may or may not be truth. So it's not really truth serum. It's just, those are just answers to questions, but they may or may not be truth. And then antimycins, very toxic compounds that are secondary metabolites produced by Streptomyces bacteria, inhibit cytochrome C reductase. So they inhibit complex 3. And finally, small molecules or ions, carbon monoxide, azide anion, and cyanide anion, inhibit the enzyme cytochrome oxidase, or complex 4. So this completes our lecture on electron transport.